So before I get going, are there any questions on the DNA viruses? Because I want to contrast them with the viruses that use RNA as their genetic material. Okay, so I'm going to start the RNA viruses again by trying to give an overview to give you a feeling for why having RNA as your genetic material causes such different kinds of viruses than having DNA as your genetic material, because there really is a huge amount of difference between the viruses that use DNA and RNA as their genetic material. But if you understand it, there's a common sense to it. Okay. I'm not going to talk about that funny hepatitis delta virus, which isn't a true virus. What I'm focusing in here are the true viruses that use RNA as their genetic material. Okay, so strategies, again, the overall viewpoint of these viruses. Now, you've got RNA as your genetic material. So what can you do? If you want to copy RNA, what can you do? And if you think about it, you can copy RNA into more RNA. So if your genome is the Watson strand of RNA, you can copy it into Crick, and then you can create Crick and copy it back to more Watson, just as you standardly do making complementary DNA. If you're doing that, you're using an RNA polymerase, but our RNA polymerase uses DNA as its template. If you're copying an RNA genome, then the RNA polymerase that copies that is going to be an RNA-dependent RNA polymerase. So when you're talking about RNA viruses, when we're talking about the polymerase in RNA viruses, it's very important to stress whether it's an RNA-dependent DNA polymerase we're talking about or a DNA-dependent DNA polymerase. So if we're going to copy everything at the level of RNA, it's going to be an RNA-dependent RNA polymerase. Or we can copy RNA to DNA, and then we can replicate DNA the standard way we always do. So this is an RNA-dependent DNA polymerase. Now, usually we copy DNA into RNA, so this is copying RNA into DNA, so it's the reverse of the usual, so it was called reverse transcriptase. And then you replicate at you can replicate at the DNA level. And if you remember, of course, the host cell, it copies DNA to RNA. It doesn't do RNA to DNA. And it does it by a DNA-dependent RNA polymerase. So it won't do this because this is an RNA-dependent RNA polymerase. And there's a lot of difference between a double-stranded DNA template and a single-stranded RNA template. And our RNA Pol2 will not copy these viral RNAs. Okay, so we don't seem to provide the enzymology for these RNA viruses to copy their genome. So at least in terms of humans and mammals, we don't provide any of this molecular mechanisms. So this means that the viruses have to code for it. So if you're an RNA virus, since we don't provide any enzymology for RNA viruses to copy themselves, they have to provide their own. So straight off, before you think of anything else about an RNA virus, you know that it's going to have to code for an enzyme to copy its RNA into something, because we don't provide that sort of enzyme. So all of these viruses code for an enzyme to copy their RNA into something. And in some cases, it will be an RNA-dependent RNA polymerase, and in some cases, it will be an RNA-dependent DNA polymerase, which is also known as reverse transcriptase. And you see that reverse transcriptase in the retroviruses, but there are some other viruses that use RNA being copied to DNA, and one of those is hepatitis B virus, which is why some of the anti-HIV drugs are also active against hepatitis B. Okay, it's a DNA virus, but it actually has an RNA stage in its life cycle. Okay, so all animal RNA viruses code for a polymerase. It will be an RNA-dependent polymerase, and whether it makes RNA from that or DNA from that will depend on which virus you've got. Okay, so now we go back to discussing the logic of how you do things when you can't use the host cell. So, for these RNA viruses, the fact that the host cell has got a DNA-dependent RNA polymerase, otherwise known as Pol2, is irrelevant because it can't copy RNA. So when they get into the cell, their RNA that gets into the cell has to be copied by their polymerase. Now, what happens with these different types of genome? And I said in the classification stuff that it's really important for these viruses which type of RNA they carry, because it affects, well, which kind of nucleic acid they carry and also which type of RNA they carry, because it affects their life cycles. So here we've got a virus which is packaging positive sense RNA. By definition, positive sense RNA is the same sense as message. In other words, this is stuff that if it sees ribosomes can be translated into protein. So if you're a virus that packages positive sense RNA, and many viruses are, that includes the picornaviruses, which includes polio, they just basically inject their positive sense messenger RNA into the cytoplasm, and then it can be translated. And then it can make all these viral proteins, and they can replicate the RNA and go ahead and do the life cycle. So that's simple. So the RNA, the positive strand RNA viruses have a pretty simple life cycle. You put the RNA into the cytoplasm, and it goes. And if in the lab, if you've got some of the RNA and you inject it into the cytoplasm of a cell, you'll get virus particles coming out. So it's a pretty easy life cycle. But what happens if you put the opposite sense? So you put negative sense. Since message is positive sense, it's called negative sense or minus sense. So if you put negative sense, and in these lectures, I've tried to put negative as blue and positive as red. So if you put a negative sense RNA into the virus, into the cell, this fried egg thing, by the way, the yolk is intended to be the nucleus. So this is all going on in the cytoplasm because there's no point in going to the nucleus to make your RNA. There's nothing there for you. So if you put the negative sense RNA into the cytoplasm, it has to be copied into positive sense message before you can make any proteins. And since you can't make any proteins until you've done this, you need to take the protein to do this with into the cell with you. So this is an RNA-dependent RNA polymerase, and it has to go into the cell with you, so it must be packaged in the virus particle. So for these viruses, they have to take the RNA polymerase in with them. So if you extract and just take the RNA from these and put them into the cell, because they don't have an RNA polymerase, they can't make message, they won't go anywhere. So it's kind of like I talked with the pox viruses. If you need to take a protein in with you that's essential to the life cycle, then if you inactivate that protein or remove it, you're not going to go anywhere. So that's what will happen here. So it has to package the RNA in the virion, which means that this negative sense RNA is not infectious unless you also give it this protein. Also, I said that to work well, messenger RNAs have to be capped, methylated, and polyadenylated if they're going to be reasonably stable and translated efficiently. Or they have to have some other features which are going to take over for that. And by and large, most of these viruses have decided to take the easy way out. They basically cap, methylate, and polyadenylate. They don't use our enzymes. Our enzymes are in the nucleus. And the other thing about our enzymes is they go around in a sort of mini factory with host cell RNA polymerase II. And it's like a factory assembly line. So the RNA, as it were, is made by RNA polymerase II, and it feeds in at one end of RNA polymerase II, and then it feeds out past all these other accessory enzymes that are going to do the modifications. So in the host cell, in the test tube, we can separate them out and they'll do them individually. But in the host cell, this is a kind of little organized factory, very efficient because of that, so that the RNA is automatically capped and methylated and polyadenylated as it comes out of this complex. So because it's a complex, if the complex doesn't get to make the RNA, it doesn't cap or methylate or polyadenylate it either. So it's no good for it. So these have to make in, if they're going to cap and methylate and polyadenylate, they have to include those enzymes in the virus particle, and they have to code for them themselves, because our enzymes like this will just go to the nucleus. So these have to be coded for. So these viruses have to code for everything to make their RNA, because they're RNA. If they were DNA, they could pirate some of our stuff. So any questions on that? So positive sense, easy, you just go in, you make it. Negative sense, you have to take all these enzymes in with you in order to do it. But both of them have to provide their own enzymes because we don't help either of them. What about double-stranded RNA genomes? You'd think they'd have the best, because you think, well, one strand's message sense, so the message sense can be used as message. It turns out ribosomes are pretty wimpy things, and they will not unwind a double-strand RNA. So the double-strand RNA is not a template for protein synthesis. So if you've got double-stranded RNA and you're not a template for protein synthesis, you have to make some message, which is. So you need an enzyme that will copy double-stranded RNA and spin off single-stranded message. So that's very like our polymerase, except our polymerase copies double-stranded DNA and spins off single-stranded message. This is going to be copying double-stranded RNA. So it's the same problem that the negative viruses had. It has to take this enzyme in with it because it can't make it until it's made the message. So it has to take it in. So these contain an RNA polymerase. Their RNA polymerase is in the virus particle as well. And again, if they're going to use modifying enzymes, they also have to be in the virus particle so that the first messages they make are good, solid messages which are efficiently translated. And then all of these, once the messages are made, they'll make lots more of these proteins and everything can get going. But it's getting things going that's critical. Okay, so those are plus, minus, double-stranded. There's one last category I want to include, and I'm not going to be talking about these anymore. My husband is going to be talking about these. These are the retroviruses. And then the retroviruses, they actually package plus sense RNA. So if it got to see ribosomes, it would make good proteins. But the way that the life cycle of retroviruses work, it does never get to see the ribosomes, so it never gets to make the proteins. Because everything in the retroviruses remains inside the nuclear capsid till they get to the nucleus. So they never get it, the RNA doesn't get into the cytoplasm to make message at this stage. So you've got a positive RNA in the virus particle, it's converted to double-stranded DNA in the virus particle, and then it's delivered to the nucleus where the double-stranded DNA gets into the nucleus. And what happens after that, you can wait for Richard. But here then, this is reverse transcriptase, and because this RNA never gets free of the nuclear capsid, it's kept protected there until it's been properly copied and everything. Because it's kept inside the nuclear capsid, it can't be translated in the cytoplasm, so you have to take the retrovirus has to take the reverse transcriptase in with it. So it also takes it. So actually, the majority of these viruses have to take all the enzyme machinery in with them. The only people who don't, or the only viruses that don't, are the positive strand viruses. So when we look at viruses that don't go via a DNA phase, in other words, everything except the retroviruses, the plus stranded do not have to put the polymerase in the virus particle. They still have to code for it because we don't provide it, but it doesn't have to be in the virus particle because the message that they put into the cytoplasm can make it as soon as they get into the cell. So if you take naked RNA, it's infectious, and the initial event when it gets into the cell is going to be translation of that genomic RNA. For the negative sense and the negative stranded and the double stranded RNA viruses, they need not only to code for a polymerase, they need to take it in with them because because they need to take it in with them, they're not infectious. And the reason they need to take it in with them is because since they don't function as message, the first thing they have to do is make message. And of course, we call the process of making message transcription. So, so the transcription is the first thing, and therefore you have to take the enzyme in with you. Any questions? And retroviruses, again, have to put the polymerase in with them. And again, their RNA is not infectious. And in this case, it's because even though they are positive sense, they actually don't release the message to the cytoplasm when they infect cells. They're heading straight to becoming double-stranded DNA and getting into the nucleus. And so the initial event here is reverse transcription. So transcription is using DNA, and us is using DNA to make RNA. Their first thing is to use RNA to make DNA. Okay, any questions on that? Because as we go through these life cycles, that's going to affect everything. It also means that If you can inactivate these virus particles because they got enzymes in the virus, if you can activate those polymerases, then they're not infectious. The monocistronic RNA problem raises its head in RNA viruses, and we've already gone through that this morning. So the business that the ribosomes load on at the cap methylated end and then fall off after the first gene. So if there are any genes further on down here, they're never going to be translated. So what do RNA viruses do? Well, you can make one monocistronic RNA per protein. So every gene has got its own promoter and it makes its own message. And that means you've got to have a lot of promoters and a lot of signal sequences, and you can do it, but they don't necessarily do it. If they do it, they tend to have some tricks to do it, which explains some of the oddities. Or they can do just what we talked about with SV40 and adeno. They can make a primary transcript and then do a lot of splicing to generate a lot of messages. So just like that single late message for adenovirus, which was cleaved down so that one after another of the other genes could be brought close to the start codon, in different RNAs, of course. But, you could do that. If you're going to do that, you've got to go to the nucleus, because this takes a lot of machinery to do, a lot of coding potential. So if you're going to do that, you have to go to the nucleus. And that's probably one of the best reasons for going to the nucleus, to use the splicing machinery. It will use these RNAs that they make, but the point is, you're not going to get much else from going to the nucleus, and most of the RNA viruses grow in the cytoplasm. There are a few that go to the nucleus, and not surprising, the few that go to the nucleus have splicing. But most of them do not have splicing, they're cytoplasmic. Or another way you can do it is you can make a large protein and cut it into smaller pieces. Now we do that. We make precursor proteins from which we clip various things. So, for example, some of the endorphins are clipped from a large precursor that you can clip endorphins from. We do it for other proteins. Not terribly frequently, but again, viruses take these things and use them. So you can make a large protein and then cut it into smaller pieces, each of which functions as an independent protein. And then you can make all your proteins that way. That way, you need to make one promoter, one messenger RNA, and you just make all the proteins strung together and then clip them off. These proteins are called polyproteins, and a lot of these viruses do it. And that's part of the reason you see so many polyproteins, because it gets around the idea. Instead of making a blue, purple, and a purple protein, you make a blue-purple protein and then clip between it. So you wouldn't have this bit in between. It would just be immediately following. Or you can actually imitate the bacteria in a way. Bacteria have these Shine-Dalgarno, do they still call them Shine-Dalgarno sequences in your thing? Which enable the ribosomes to be recruited to the start of each to the region where a protein is going to be made from, to the start codon region. You can actually do that for eukaryotic ribosomes. It's not a Shine-Dalgarno sequence. It's a kind of elaborate folding. It takes, in the case of poliovirus, which uses it, it's probably getting on for 400 or 500 nucleotides. And it folds into this elaborate thing that they call a pseudoknot. And so it's an elaborate three-dimensional structure, like tRNAs are elaborate three-dimensional structures. This is also a very elaborate structure. And apparently it just spooks out the ribosome into thinking it's a real start site, and it will bind to these. So you can actually make it so that you can bind here rather than here. When this was discovered, everybody said, great, viruses are doing this thing, and the rest of us are going, viruses are never that special. We have genes that we do this on. In fact, a lot of the genes that see us through tough times do this. And part of the reason we do that is so that we can activate cap-dependent methylation, cap-dependent protein synthesis in tough times and just basically keep everything ticking over. And so we actually seem to have these sequences so that genes that are made in really tough times can be made in the absence of all the others. So it's a way to sort of code genes as to what we need to keep going when times get tough. So we have these, and a lot of our genes have them, particularly quite a lot of these things like growth factors and things which are involved in seeing us through stress. But these viruses can use it very professionally. Okay, so one of the variations that we're going to see is because viruses have the monocystronic problem. So a lot of these things that look like they're doing everything under the sun means they are doing everything under the sun, but it's just to get around the problem of how do I make a monocystronic RNA, especially if I'm in the cytoplasm and I don't have access to splicing. Another thing I want to point out that colours having RNA as your genetic material is the genome size of these viruses. These viruses are all going to be pretty small genomes. They may be large virus particles, but they'll be pretty small genomes. And I just want to go into why that is. Have you been told about tautomers and tautomerization in biochemistry? We've got a few nods, unless people are just about going to sleep. So to be brief, and I think I've got a slide in the genetics level, but to be brief, every base spends most of its time looking as it does in the standard Watson-Crick model of DNA. But every base spends a very tiny period of its time in a form where the electrons are slightly differently distributed, and then it so happens that it base pairs as if it was the other member of the pair. So A will base pair as if it was G, C as T. That's just a chemical feature of the bases. There's nothing we can do about it. So there is inevitably mutation going on because when a polymerase incorporates a base, if it's A behaving as G at the second that it's incorporated, it will be incorporated as if it was G. And then you look a few seconds later and it's A. We have proofreading in DNA polymerases that looks back and thinks, oh gosh, I just incorporated G and it should have been A. Let's take it off and replace it. And they actually got the machinery for it. We also have a pile of machinery that trolls along the new strand, compares it with the old strand, and where it doesn't match, where it's not complementary, cleans it up. So we have a lot of this. But we don't have it for RNA. RNA is the disposable. We make RNA, it goes to the cytoplasm. We don't care what happens to it. It can be degraded, we just make more. So we don't do it for RNA. So the point is, tautomerization means that however accurate an RNA polymerase is, it will mess up. There is no way around it, unless it's got proofreading. And we don't have RNA proofreading, and it doesn't seem that the RNA viruses have developed a good proofreading mechanism for themselves. So they've got nothing of ours they can snaffle, no genes of ours they can pick up and modify to their own purposes. They seem not to have proofreading. So any mistake they make stays. And the net result of that is RNA viruses have very small genomes, because it turns out that this tautomerism gives you an error rate of about 1 in 10,000. So that's about the limit. So if your error rate is 1 in 10,000, if you have a virus genome that's 10,000 long, statistics will tell you that if you take 100 of those genomes, that if you have an error rate of 1 in 10,000, actually 66% of them will have one error, and of the remaining 33% or so, half of them will have 2 errors, and half of them will have non-errors, which means if you make 100 genomes, you'll get about 12 or 13% which don't have an error. That for an RNA virus is perfectly acceptable, because if you're making these huge numbers, if 10% of them are good, that's fine. But you get a lot of these mutations. So if you're much larger than 10,000 nucleotides, herpes is 200,000. Herpes would have an average of 20 mutations, and most of these mutations, it turns out these viruses are so precisely adapted that they're either lethal or they mean they're not competitive, they don't do well in some part, they don't hide out well, they don't transmit well. They have some problem at some stage in their life cycle. So the net result is that most RNA viruses are pretty small. In terms of genome size, they're around the 10,000 mark max. So they're down there more in the between SV40 and adeno rather than up in the big league. So you're not going to get these huge complicated genomes that we saw with DNA viruses. And what genome they have has to code for a polymerase. Not only does it have to code for a polymerase, it has to code for a viral attachment protein. That's the only way they're going to be able to get into the next cell. The polymerase may even have to code for a methylation, capping, and polyadenylation. So by the time you've done all this, there's not much room left for anything else. So the capsid protein, of course, will have to be coded for as well. And so there's relatively few other proteins. So these viruses have very few proteins, small genomes, feature of the RNA viruses. So, but the big feature is they do some funny things to get around these problems, and that creates the weak links that we can potentially go after. So, as I say, the virion may be quite large, but the actual nucleic acid in it is pretty short. Any questions on that overall strategy for RNA viruses? So what I'm now going to do is I'm going to start off by going through positive sense, negative sense, double strand. Positive sense viruses, we're going to be talking about picornaviruses. Those include rhinoviruses, which are actually the major cause of the common cold. But since the common cold is relatively mild, we're not going to be we'll spend comparatively little time on rhinoviruses. But they also include enteroviruses, and those include things like polio and hepatitis A virus. So there are some more serious viruses that we will be talking about in the picornaviruses. Pico means very small, and RNA means guess what? And we'll also talk about the toga viruses. Toga means a cover, and so they're enveloped viruses. And flaviviruses, flavy means yellow. These are the ones that cause yellow fever, West Nile. These are flaviviruses. Okay, I'm going to just talk about the life cycle for picornaviruses here. If you understand the logic of the various things that I talk about when we're doing these RNA viruses, even though these viruses have got rather different life cycles, I hope you wouldn't find them that unusual, that awkward to understand, because they're using the same principles. But let's talk about polio or picornaviruses. Okay, they're very small, given them, hence the name. They're icosahedral. Here's a penton with 5 subunits, and here's one of these hexons on the face. This is a sort of pseudo-hexon, and it interacts with 6 subunits, 3 other, but it sort of looks like it's the symbol for the Isle of Man, and I don't know why that should be, but anyway. One thing to notice, because we will come back to talk about this later on, is this is the penton, and it's sticking right up, but around it, this is actually depth cued. So this is like some of those maps you see. The depths of the ocean are dark blue, and the heights of the mountain here are yellow. We don't have any snow. But this is depth cued to give you some idea of the depth. But these are quite deep pits on the surface. They don't go on the inside, but they're deep pits on the surface. And it seems it binds to its receptors inside these pits, and we'll come back to why that's important, particularly in flu. This tendency of viruses for the receptors to actually poke into the virus particle and not bind to the surface, but bind down into these various pits. And here it's a very large one. Penetration. The attachment protein binds inside those pits and puts the message into the cell. And it actually seems to create, it undergoes a reorganization when it binds to the receptor. It seems to create a channel that's big enough and hydrophilic enough for the RNA to get through, and the RNA gets into the cytoplasm. It's not actually capped. It has a protein at the 5' end called VP because it's in the virus particle, G because it's linked to the genome. So VPG is actually attached to the 5' end, and that seems to stabilize the RNA enough, along with other features. We'll come to probably what the function of VPG is a bit later on. But it's called VP because there's one molecule in the virus. But it's a structural protein, as always. Okay. So how does it make its message? How is its message translated? Because its genome is its message. So the genome is the message for poliovirus. And it doesn't have the 5' methylated cap structure, so it's not seen, that's not how the ribosomes recognize it. It's got one of these special sites that trick the ribosome into thinking this is like a 5' end, but it isn't a 5' end, it's this elaborately folded RNA. And this is called the internal ribosome entry site, or IRES. However, it's more frequently known as a landing pad. So the IRES is a way that you can change an RNA so that instead of having to be recognized at the 5' end, the ribosomes can actually bind internally. And actually, we make quite a lot of use of that in various of these recombinant DNA constructs that we make. So the ribosome actually, the small subunit binds to the IRES here, and then it will roll along until it finds a start codon, and then it will assemble with a 60S subunit to make an 80S ribosome, and then it will start translating. So the rest, once it binds here, the rest of the story is very similar to if it had bound here. It's just that it's able to bind here. As I say, that takes a bit of coding potential. It's not like 5 nucleotides that enable it to. It's actually a secondary structure that it recognizes. And it makes a polyprotein. So you make one long protein, it starts here, it's about 500 nucleotides in because the rest of this is taken up with making this structure, and it stops here, close to the 3' end, and you make this large polyprotein. I just wanted to point out, this explains some of the things that these viruses can do to us, because the virus is also looking for a weak link. And it's saying, whenever I do something for myself, I can target the host doing it, and I can turn it to my advantage. So the viruses are also weak link oriented. So in an uninfected cell, the host mRNA is capped, and the initiation factors, and did I say 30S ribosomal? I meant to say 40S ribosomal. The initiation factors bind along with the small ribosomal subunit, which is 40S in eukaryotes, and you get a complex which rolls along until it finds the start codon, etc., etc. What a lot of members of the picornavirus family do is they actually have a protease that will cleave one of the factors that's involved in recognizing the cap, so it can't recognize the cap. And so that factor cannot recognize a cap, and the host messages are no longer being translated because it can't recognize the cap, except for those few host messages that have an internal landing pad. But for the majority of host messages, which rely on that 5' methylated cap structure to recruit the small ribosomal subunit, they can't do it because the factor that recognizes them is in 2 pieces, and it doesn't know they're there. So this means that these factors and this small subunit are now available primarily for the use of the virus. So it's already got rid of the host cell making proteins and using all that valuable tRNA and energy and amino acid precursors and everything else, and they're all coming for the virus. So this is a way, so by not using a cap itself, it can actually target the host cell cap. For the virus, since the factors recognize the internal landing pad, it doesn't make any difference because it turns out the altered factor can still recognize the internal landing pad. So in these virally infected cells, we see primarily host cell virus proteins being translated, and a few of ours which have the internal. So it's a neat way to get your own back. So when you do something separate, you can afford to inhibit the equivalent that the host cell does. So as small as this is, it's got some ways to get back at you. Okay, so we get this polyprotein made by translation of the incoming message. And the amino terminal to C terminal, of course, it's made in this direction. And it's cleaved by a series of proteases. And I'm not going to ask you which proteases do what. But the point about these proteases is they are actually virally coded proteases. So that is important. Because if we didn't have a good vaccine for polio and hepatitis A, another way would be to try and target the proteases so that they didn't cleave it. Because if you don't cleave this protein, you're never going to be able to assemble infectious virus particles. Because we have a good vaccine, that approach hasn't been followed. But it's a weak link. So it's translated into a polyprotein. It's cleaved by its own proteases. And some of those proteases are actually active while they're a part of the polyprotein. So it's actually cleaved as it's being made, because once you get past one region, that region will fold up, become a protease, and cleave itself off. So in fact, in the infected cell, you rarely find the full-length polyprotein. It's always cleaved as it's made. But there are several proteases that are involved in these various cleavage steps. So it cleaves it down to give all the final proteins that it's going to need. As I say, it's pretty small. This end, see, they've all got VP on them. These are the capsid proteins that are going to be assembling that capsid. This end is going to be largely the proteases that are needed to do all this, plus the polymerase, because you have to make RNA off an RNA template, so it has to provide the polymerase. And it also got this protein here that's called VPG, and we'll see where that comes in a second. But that's going to eventually end up in the virus particle. So just simple self-coded proteases. Replication, just pretty straightforward. You've got positive sense RNA that's put into the cell. It's copied into negative sense RNA and then copied back into positive sense RNA. So you keep making... So the new positive sense RNA can either be used to make more RNA, or it can be translated into protein, or it can be packaged in the virus particle for the next thing, next round. The VPG is always on the 5' end, and we think the reason for that is we think this virus actually uses an RNA primer rather than initiation de novo. So it looks like the VPG story is rather similar to the terminal protein story in adeno. So it's always at the 5' terminus, and we think that's because of the role it plays in replication, as opposed to, it's not necessary for protein synthesis. You can actually cleave it off and it's still translated. So the VPG seems to be important in replication, and it's also in packaging of the virus. But so it seems to function as a primer. So usually you don't think of RNA polymerases requiring a primer, but in this case it's using a primer. And it's one way to get a protein on the end. It's going to need some way to protect this in this infected cell, and the VPG seems to be reasonable, because that's what our capping and methylation do. Most of the RNAs that are stable in the cytoplasm have got secondary structures or things on them to protect them against being degraded. So the viral RNA polymerase is often called a replicase because it's involved in replication. But of course, one of the things it does when it's making replication is, since it makes more plus sense strands, it's also making more messages. So, it's often called a replicase. And I just want to stress that these polymerases do have several names because they produce several functions. So anyway, that replicase makes the copies of the RNA, and it uses host cell factors as accessory proteins. But interestingly, the host cell factors it uses are factors that are present in almost every cell. So that doesn't limit its host cell range, but it does use some host cell factors. These are host cell factors that do other things in the host cell, but it's just found that it can bind to them and use them for its own purposes. And I've already said about what happens to new plus strands. You can just keep the cycle going with them, or you can spin them off to become messages, or you can package them to become virus particles. So how do we assemble these? If you looked on that picture of the polyprotein being processed, and I don't expect you to remember this as in terms of the names of the things, but there was a protein called VP0 that was one stage above the bottom. The virus capsid is actually assembled with VP0 plus VP1 and VP3. And at this stage, it's a little bit not terribly solid. And you can get now the VP RNA, the RNA with VPG on the end, can go in. And it looks as if it likes to package RNA with VPG on the end. And that makes sense. This has been a nice way to tag your RNA so that your RNA, the virus particle, packages the RNA that corresponds with poliovirus RNA. Otherwise, if it packaged every other RNA, you'd be having, it would be like generalized RNA transduction. You'd be packaging all these individual messages. So the fact that you have a different end to the message may very well help make sure that you get the right stuff in. So the VPG RNA enters, and once that VPG RNA is in here, there's a mystery happens, but it clearly happens, and that is something changes, and in the process of that changing, there's a protease activity that's activated inside the virus particle that actually cleaves VP0 into VP2 and VP4. So one of those capsid proteins get cleaved, and the net result of that is it can undergo some conformational changes, and now the whole capsid morphs into a really tight, solid little capsid that's going to protect that RNA, because RNA is very, we're covered with ribonucleases, so these capsids need to be really tough. So once the RNA is in, the hatches are put down, and that's because the RNA actually triggers the change that will cause it to become a tight virus particle. And now you see, again thing, huge excess. These viruses tend to just go in. If you look at the end of the lecture on virus growth and what these things look like in their CPE and tissue culture, the ones that were growing poliovirus, the cells were just blown apart. And that's what polio does. These cells are going to lyse. They're going to be really sick. They haven't been making their own proteins for a while. It's actually also rearranged their lipids for them so that their membranes are all different, because it actually likes to replicate its RNA on membrane surfaces. And then it's assembled tons of virus particles. You can see all of them here forming this paracrystalline array. So with these RNA viruses, I said most of them grow in the cytoplasm. So what you see are inclusion bodies in the cytoplasm. So inclusion bodies in the cytoplasm tend to, for viral infections, tend to indicate an RNA virus or a pox virus. And then the cell will lyse and release all of these into the environment, and not all will have RNA, and given the error rate, a lot of those RNAs will have mutations in and not be able to establish an infection, but you've got so much excess that you'll produce many infectious particles from one cell. So are there any questions on poliovirus? Pretty straightforward, it's not. Okay, so that was the positive sense. And the other positive sense are sort of variations on a theme. But again, when you go to the other families, they each have some different specialities slightly. And again, those are potentially weak links. The problem is, with bacteria, a weak link is that they've got the 30S70 as opposed to the 3060 as opposed to the 4070. I'm getting so confused this morning. They have different size ribosomes than we do. They have a different method of protein synthesis than we do. They have cell walls by and large. So we have a whole pile of targets that if they work on one bacterium are likely to work on a lot of bacteria. We just don't have that for these viruses because of that. So while we can potentially target each of these weak links, since they're virus specific, it really has to be economically valuable in order for pharmaceutical people to go off and do it. By and large, a vaccine is much better. You prevent having the disease rather than treating it. And vaccines tend to be easier approach than doing all the clinical trials for the various therapeutic agents. But with the advent of things like HIV and so, we're developing a huge pile of protease inhibitors, nucleoside analogues that can potentially be used to target some of these things in viruses. We just have to screen them and check them out, and that has to be economically attractive. Okay, non-segmented negative strand viruses. Okay, so we're going to negative strand viruses, and there's a bit of difference between the ones that are segmented and not segmented. And if you remember, segment just means, is your genome in one piece, in which case you've got one chromosome or you're non-segmented, or is it in multiple pieces, in which case, if it was humans, you'd say you'd have multiple chromosomes. If they're bacteria, you say you've got multiple segments. I guess we don't want to admit that they have chromosomes or something. I'm not sure why we use segments, but we do. Anyway, we've got these families. I'm going to talk about all of them, but I'm going to start off by talking about the rhabdovirus family. Rhabdo means rod, and I hope you'll see why in a minute. Okay, so rhabdoviruses are rod-shaped like this. In fact, more frequently they're called bullet-shaped, but they're basically rods. So they're kind of like a microbacillus. And they are enveloped. And this is a case of, they are the slinky, which has been shrink-wrapped. So when you look at the virus thing, you look at something that looks like it's a shrink-wrapped slinky when you look at it in the EM. This is a cross-section through it. And I've tried to use the same terminology. So the genome is blue because it's negative strand. The green colour here is the bilayer lipid membrane. And then it's got glycoproteins inserted into it, just one glycoprotein, multiple copies. That's going to be the attachment protein. And it's also going to be the protein that triggers fusion, because being an envelope virus, it has to fuse either with a cell membrane or with an endosome membrane. And since it's not a paramyxovirus, a herpes virus, or HIV, it's going to freeze with an endosome membrane. So even if I didn't ask, you should be able to tell me that this fusion protein is going to be active at acid pH. Okay, and underneath that, there's something called the M protein. And M can be thought of as standing for maturation. It's involved in enabling the nuclear capsid to recognize the glycoprotein, because it binds both to the cytoplasmic tails of the glycoprotein and to the nuclear capsid. So it's a bridging protein that brings these together during maturation. And we'll see this kind of way of having a bridging protein you see in a lot of these other envelope viruses, including retroviruses. They don't have to be related viruses. It's a general principle of this kind of structure, and helical. And any helical RNA virus is going to be enveloped. So in mammal, the ones that you're going to be concerned about, the ones that cause human disease, they're all enveloped. Okay, so its negative sense, we've already said you need to make RNA. So we've already said that because of that, when it gets into the cell, you need to copy the negative into positive. You need an RNA-dependent RNA polymerase that you have to code for. And since you can't make it until you've made a message, you have to take it in with you. So it's in the virus particle. And in fact, if we go back there, oh no, I need to go forward. It's in the virus particle associated with the nuclear capsid. So that's where it's going to be working, and it binds to it. This virus takes the approach of, you might as well, if you're either for them or against them, this one takes it for them. So it's capped methylated, polyadenylated. So all of that has to also be in the virus particle. So already it's actually taking more than half of its genome up with making its messages. Okay, and I should have said, one of the viruses in this group, the really significant one from the point of human disease, is rabies. But there are other diseases that are animal diseases, but humans can get them, but they tend to be pretty mild. So the one that we're really concerned about with this one is rabies. But I used to work on this family, and I worked on one that you get in cattle called vesicular stomatitis virus. It gives them vesicles around their mouth. It infects other farm animals as well. And so, again, you work on what's safe rather than the real thing. Your idea being that once you've worked out what's going on, then the people in the real safe laboratories have got some idea of what experiments to do, and they're not just floundering around. So the polymerase complex is associated with the nuclear capsid. And the nuclear capsid, it's helical, and the N protein, N for nuclear capsid, is what coats it. You may, in some books, people are trying to get people to call nuclear capsid proteins NP. The historical name here is N, and they're changing slowly. Okay, so this is what I said about it being a shrink-wrapped slinky. Here you can see the outside of the slinky, and there's a suggestion, at least from where I'm standing, of the bands across at the edge of the slinky. Here, the membrane hasn't been perfectly intact. The dye is penetrated, and you can actually see the outside of the slinky and the hollow center. So it really is like this kind of slinky molecule. And then this is the membrane around it. And if you look very closely, and I doubt you can see from where you are, but if you've got a computer in front of you, and maybe on the handout, I don't know, you can see there's a fringe here that looks like the pile of a carpet. That one's got a very clear one. Some of the others are not so good. And that is this spike protein, this glycoprotein that's in the membrane, and that's the attachment, and in this case also the fusion protein. So vesicular stomatitis virus is the one that everybody works with, and then a few people apply what you learn from that to rabies. Okay, binds to a receptor on the cell surface, taken up by endocytosis, acidified. Under acid conditions, that glycoprotein undergoes a shape change and can now work as a fusion protein. The nucleocapsid is released into the cytoplasm. Now, it's a helical nucleocapsid. So how do you get the nucleoprotein off the RNA? And the answer is you don't. Because I told you that ribosomes are wimpy and they're not very good at separating two strands of RNA to find out which one is message. They also don't actually take off this nucleocapsid. But the point is, this nucleocapsid, what's it going to do? It's a negative strand nucleocapsid. It's going to be used as a template for RNA synthesis. The RNA polymerase is going to be the viral RNA polymerase. So there's no reason why it should work the same way as our host cell RNA polymerase. It can actually work and copy the RNA while it's part of this helical nucleocapsid. So remember I said, oh, I forgot the shoes. Oh, well. But if this is the RNA, it would be coated with shoes. But if you have it so that the polymerase can get in and look and see the order of the nucleotides, but RNases can't get in because you've protected the phosphate backbone, then you can copy this from your polymerase without getting out of the nucleocapsid. And since that's the only thing that this is there for, to make the new plus sense RNA, as long as the polymerase can see you when you're in the nucleocapsid, you don't need to uncoat. So although in a way you're a bit more vulnerable outside because you've got to keep your polymerase active, once you get into the cell, you're a bit less vulnerable because your family jewels are protected by this nucleocapsid protein. And it is really good. I mean, even if you take it outside the virus particle and add ribonuclease, it will stay intact. So this is a very good coat. So the nucleocapsid remains on the negative strand RNA viruses. They're not going to be translated by proteins. So the positive strand RNA viruses have to be brave and handle their RNA and have it going into the cell and not remain in the nucleocapsid. But the negative ones can keep it in the nucleocapsid. Okay. It's going to replicate in the cytoplasm just like the picornaviruses did, nothing from the nucleus. And the genomic RNA will remain, as I say, in the nucleocapsid form. So what happens? Well, you've got this genome coated with nucleoprotein, and you've got a polymerase attached to it, and you want to make message. So we call the process of making message transcription. So sometimes we call this RNA-dependent RNA polymerase a transcriptase because it's going to make RNA. And it makes individual messages by a mechanism which is not entirely clear, but seems to be a way to not spend too much effort on having big elaborate promoters. So it makes one message for each of the proteins. It happens to code for 5 proteins, so it makes 5 messages. And this really big message is the polymerase, and it also includes polyadenylation, capping, and methylation activities as part of the polymerase. In our cells, those are separate proteins, but in these viruses, they're frequently a part of the same complex, and in this case, part of the same protein. So it caps, methylates, and polyadenylates because just like the host cell polymerase, as it makes the RNA gets capped and methylated and polyadenylated as part of that factory assembly line. You can't just give it an RNA and say, here, cap and methylate it. It does it as part of the assembly line. Okay? When these, you're in the cytoplasm anyway, so they don't have to be shipped. When these are translated, they're translated into protein. And when there's plenty of that protein, you now go into replication. And that seems to be because as you go into replication, the protein actually coats. This is in a nucleocapsid form. As the protein is made, it coats the growing plus strand. And that switches you over from making individual messages to making a full-length copy. Same enzyme, so now we call it a replicase, same enzyme. You make a full-length copy of the plus strand, which is coated with nucleocapsid and kept protected, and then it's copied into negative RNA, and that's also coated as it's made. We'll talk about the rest of that on Monday. Are there any questions?